Dear student, today we are going to present the scientific journey of Professor Jens Christian Skou. Who is Professor Skou? Jane Christian Skou was a professor emeritus at the Department of Biophysics of the University of Aarhus, Denmark. So why we are so much interested about him? What did he do? He received half of the Nobel Prize in Chemistry in the year 1997 for his remarkable discovery of sodium potassium ATPase pump. Now, his story tells us how the scientific curiosity, persistent work can bring a medical doctor working in an isolated place like Denmark University to a major discovery in the field of biochemistry. And very few people knows that initially Skou was actually trained as surgeon and wanted to understand the mechanism of local anesthetics. This actually led him towards the area of research. Now Skou studied in the medical school in the Denmark when Denmark was under German occupations during the time of Second World War. This period has a profound impact on his life. Many of Scouts part in the resistance against Germans and they went underground. So this is the Aarhus University during the World War II. Many of his teachers they went underground, escaped to Sweden. So many times their classes remained cancelled. However, he completed his graduation in this hard situation and started internship in the northern part of the Denmark. But surprisingly and interestingly, in the surgical ward, he had to take the responsibility of a surgeon despite being an intern. As all the senior professors, they took part in the war against Germany. Now, this young Skou actually started his work, research work, in the beginning of 1950. At that time, few things are known that intracellular space has high potassium, extracellular space was high sodium, and in the year 1939, it was also shown that muscle membrane is permeable to sodium, and it was suggested that there are pumps in the membrane. Okay, so when Professor Skou was starting his research, existence of pump was existed, but nobody knew about the nature of such pump. And Professor Skou knew that he was interested about the drugs, local anesthetics and anesthetic drugs. He knew that narcotic drugs, there was a relationship between the narcotic drugs and lipid solubility, narcotic drug potency and their lipid solubility. Actually, and the local anesthetics, he knew that they were the weak bases which in aqueous solution dissociates into water soluble ionized part and a non-ionized part which is lipid soluble. So local anesthetic also has some lipid solubility. So did a similar correlation exists between the lipid solubility and the anesthetic potency of the local anesthetic? It was his question. But what happened? He found that there is no, during his initial experiments, he found that there is no correlations there was between the capillary activity and the potency of the local anesthetics. And at some point, Skou realized that a phospholipid monolayer is similar to one half of the cell membrane. So he found that these local anesthetics, they don't have any correlation between the capillary activity and they could not penetrate this lipid bilayer. But when he observed that when he is using lipid monolayer, when he used this lipid monolayer to test the effect of local anesthetics, he observed that local anesthetics, when added to the water phase underneath a monolayer of lipids extracted from peripheral nerves increase the pressure in the monolayer with a given area. That indicates what? That indicates that they penetrated up to the monolayer. And there was a correlation between the ability to increase the pressure in the monolayer and blocking potency of the local anesthetic drugs. These were his observations, and on these results he published 6 papers in English. And at this point, he could have stopped research like all other scientific scientists, glorified popular scientists. But his curiosity did not let him stop. He wanted to determine what the connection was between the penetration and pressure increase in the monolayer and the blocking of the nerve impulse. The local anesthetic in blocking concentration had no effect on the membrane potential. So they are not changing the resting membrane potential. So Skou thought that it was possible that these local anesthetics, by increasing their pressure, they're blocking the opening of some membrane sodium proteins. But how to prove that? This was a problem at that time because there was the protein responsible for opening of the nerve membrane for sodium, it was unknown, and there was no methods existed for measuring the conformations of proteins in a monolayer. So what to do? So he decided to use a protein which has enzymatic activity and incorporate it in a lipid monolayer, add local anesthetic to the water phase, and see if penetration up into the monolayer had an influence on enzymatic activity. If it is there, then he will take it as an indication of an effect on the conformation. Now for it, he required an enzyme with high turnover number, as the number of protein he could incorporate into a monolayer was very limited, and if possible an enzyme that was membrane bound is preferred, and one of such candidates is the acetylcholinesterase protein. Now acetylcholinesterase is a membrane bound protein and which is prepared actually from the electric organ of the electric eel. Skou had no access to electric eel, but he knew that David Nachmann, a former refugee from Nazi Germany, now at Columbia University, he prepared the enzyme from electric eels. And Skou read a textbook by Nachmann's son in the year 1948 that has shown that there is an ATP hydrolyzing enzyme in the membranes of the giant actions, an ATP ase. He knew that the ATP is the energy source in cells and wondered that what is the function of this ATP hydrolyzing enzyme in the nerve membrane, why it is hydrolyzing energy and ATP and generating energy. Furthermore, being in the membrane, it had to be a lipoprotein. And that was what he needed for his monolayer experiments. So he decided to have a look at the enzyme when he returned to Harahas. In September in New York, he prepared the acetylcholinesterase monolayers and measured the enzymatic activity of the monolayer and observed that enzymatic activity of the monolayer was pressure dependent. The next step was to prepare a phospholipid monolayer containing proteins and adding a local anesthetic to the water phase and see the enzymatic activity whether it is influenced or not. But for it, he required the giant axon. He had no access to the giant axon. So what we will do then? He used membranes from crab leg nerves instead. A fisherman sent him about 200 small crabs every week. and he used several thousand crabs before the study was over. We can see here Professor Skou with the crabs. Okay, so what actually happened in his lab? His lab assistants used to hammer those lab. Then these crabs were placed onto the boiling water so that they will die. Then they will cut their legs and separate these large actions. Okay, giant actions. Now, one problem was there. These procedure gave them a peculiar smell of the boiled crab, and one could easily locate the Institute of Medical Physiology on the campus of Aarhus University based on its smell alone. So, from where the smell is coming, it must be the lab of Professor Skou. Now, Scone at that point entered in a very frustrating period because the membrane pieces from the crab nerves contained, as described for the giant axon, an enzyme that hydrolyzed ATP in the presence of magnesium, but the activity was very low. And the action of addition of potassium has no effect. Sodium gave a very slight increase. Okay, and the further experiments gave, however, a varying results. So he needed an enormous stamina not to give up. Now opportunities came up for the clinical jobs. And at one point, Scott decided to take up such opportunities or appointments. But his wife knew him better and suggested that he should stay at the university. As it turned out, eventually, Skou discovered that the enzyme required a combined effect of sodium and potassium activity, and this was an unexpected surprise. The enzyme, which was influenced by combined effect of sodium and potassium, was related to this change in permeability of sodium potassium, and that it was a channel in the nerve membrane. But as the change in permeability of the nerve membrane was voltage dependent and the enzyme activity was ATP dependent, so SCO has come to a decision that these enzyme must be a part of the sodium pump. Now, with these experimental data, he published his findings in a paper entitled "The Influence of Some Cations on an adenosine triphosphate from Peripheral Nerves." It was published in Biochimica, Biophysica, and 19 in the year of 1957. Okay. Now, initially, actually, he ended the paper by saying that the crab nerve ATPase seems to fulfill a number of the conditions that must be imposed on an enzyme, which is thought to be involved in the active extrusion of sodium from the nerve fiber. And this paper was the principal basis of his Nobel Prize. But initially, this paper was not that much popular. Gradually, Skou's discovery spread, and when he visited the United States in the year 1963, the editor of Physiological Review asked him to prepare a review article on the ATPase, and it was published over there in the year 1965. We can see the paper. But it took a long 40 years for the recognition of his contribution to science. In the year 1997, he has been awarded by the Nobel Prize. So this is the incredible journey of a famous scientist, Professor Dr. Jen Christian Q. Thank you.